Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome, curious minds, to another deep dive. Today we're tackling just a fascinating new paper. It really flips a major assumption about how our brains handle inflammation.
Oh, definitely. It's a pretty profound shift in perspective, actually.
Yeah. So get ready for this.
Yeah?
It suggests that a system we, thought was a helper in immune cells, it actually becomes a kind of saboteur in our precious neurons, particularly in diseases like multiple sclerosis.
Right, MS. For years, the immunoproteasome has been understood as, well, a system that boosts immune cell activity, helps maintain cellular health. But this research asks, what if, inside neurons, this very same system turns rogue and actually drives damage?
That's really the core finding, isn't it? This research uncovers a really counterintuitive truth. In inflamed neurons, a specific part of this immunoproteasome called PSMB8 actually impairs the cell's crucial protein recycling system. It's like discovering the cleanup crew is, accidentally making the mess worse.
Leading to metabolic disaster and ultimately neuron death.
Yeah. Okay, so maybe let's set the stage a bit. Let's unpack the challenge of neurodegeneration in multiple sclerosis, or MS, because this isn't just about inflammation, right?
Not at all. It's also characterized by this relentless, progressive loss of neurons. And sadly, our current immune-targeting therapies, while they're absolutely crucial...
They don't fully stop that neuronal damage.
Exactly. That's a critical point. Neurons are incredibly delicate cells. They rely on two really tightly linked processes for survival. One is proteostasis, that's the balance of building and breaking down proteins, keeping things tidy, basically.
Right, the housekeeping.
And the other is metabolism, their energy supply. Super important. Now, when inflammation, often driven by signaling molecules like IFN gamma, hits the central nervous system, it can induce the production of this immunoproteasome. It's a specialized protein degradation complex.
And here's where the mystery has kind of been, right? We knew inflammation messed with the proteasome, the cell's main recycling plant. But how that leads to metabolic failure and neuron death in the brain? Well, that remained a bit of a black box.
Precisely. Understanding this specific connection is vital. Because, it could give us entirely new ways to protect those precious neurons.
Even if the inflammation is still going on.
Exactly.
So the researchers began their investigation by looking for very specific changes, right, inside the neurons during inflammation. What did they initially see when they looked into the spinal cords of mice with EAE, that's the MS-like condition, and even human MS brain tissue?
Well, what immediately stood out was a really significant increase in an immunoproteasome subunit. It's called PSMB8. And crucially, it was inside inflamed neurons.
Inside the neurons themselves?
Yes,
Okay,
they used advanced techniques, RNA sequencing, neuron-specific proteomics, and they confirmed this both in the living EAE mice and in human MS brain lesions. So this wasn't just some general immune response nearby. It was happening within the neurons.
Okay, but here's where it gets really interesting and kind of unexpected. Because in most other cells, like immune cells, this immunoproteasome usually increases protein breakdown, right? Helps keep things balanced under stress.
That's the standard understanding, yes.
Yet in these inflamed neurons, even with higher PSMB8 levels,
Yeah.
the overall proteasome activity actually decreased.
It's a complete reversal.
A total flip.
Yeah, and that finding was a crucial aha moment. It suggested PSMB8 might be acting, well, differently, maybe even neurotoxically in neurons. So to test this, the team used an immunoproteasome inhibitor, ONX0914, in the EAE mice. And they also created neuron-specific PSMB8 knockout mice.
Meaning they took PSMB8 out just from the neuron?
Just from the neurons, leaving other cells untouched. Very targeted.
On the results.
They were really striking. Both the inhibitor and knocking out PSMB8 in neurons significantly reduced the disease severity in these mouse models. And more importantly, it protected against neuronal loss. It preserved synapses.
The communication points.
Exactly. And axons, the nerve cables. It even improved mitochondrial health of the cell's power plants.
Wow. So it's clear that neuronal PSMB8 isn't just sitting there, it's actively driving the damage.
Absolutely.
So for you, the learner, This is a really clear signal. The cellular context matters immensely for how these systems work. What's beneficial in one cell type can actually be harmful in another.
A crucial lesson in cell biology.
Okay, so building on that initial discovery, they then looked at how this PSMB8 accumulation directly harms neurons. They basically forced neurons to express PSMB8 in lab cultures and just watched what happened.
Right. And what's fascinating here is that sustained PSMB8 expression, highly neurotoxic, it led to widespread neuron death in these cultures.
Just killed them off.
Pretty much. But crucially, they could rescue this toxicity. If they co-expressed PSMB5, that's the regular proteasome subunit that PSMB8 usually replaces, then neurons survived much better.
Ah, so it shows a direct competition. When PSMB8 pushes out PSMB5, that's when the trouble starts.
Precisely. It's not just a simple swap. It fundamentally changes the proteasome's function, its ability to clear certain vital proteins.
So it's not just PSMB8 being present, but its specific integration, kicking out PSMB5, that causes the problems. They also found, didn't they, that PSMB8 induction by IFN gamma made neurons much more vulnerable to something called excitotoxicity.
Yes, that's a common pathway for neuronal damage. And this increased vulnerability was completely prevented if you knocked out PSMB8.
So it really solidifies PSMB8's role as a key driver of damage in multiple ways.
Which raises an important question. What's the specific molecular fallout from this PSMB8-driven dysfunction? They looked at gene expression and found PSMB8 altered genes related to redox balance. That's how a cell manages damaging reactive molecules.
Like rust-proofing the cell.
Kind of, yeah.
Yeah.
And also metabolic coenzymes. And confirming this, they saw significantly higher levels of reactive oxygen species, ROS. Those are harmful metabolic byproducts in the PSMB8 expressing neurons.
More ruts.
Exactly. And they also saw a reduced expression of inducible nitric oxide synthase, another marker related to ROS production, in the mice where PSMB8 was deleted. So it suggests PSMB8 directly messes with the cell's ability to manage oxidative stress.
Okay, so for you listening, pretty direct link.
Yeah.
PSMB8 accumulation in neurons leads to more toxic stress inside the cell. Makes it unstable, primes it for damage.
That's the picture emerging.
So PSMB8 piles up, ramps up toxic stress.
Okay.
But this brings us back to that earlier really weird finding. The overall proteasome activity was actually decreasing. How does that work? How can a system meant for breaking things down slow down when a subunit that's supposed to enhance it is increasing?
Right, it's the central paradox. What's fascinating here is that they measured the activity directly. PSMB8 overexpression drastically decreased the metal-5 catalytic activity. Think of it as a specific cutting tool of both the 20S and 26S proteasome.
Those are the main machines doing the breakdown.
Yes, the core machinery. And this is a direct contradiction to its known function in other cell types. This explains why the overall proteasome activity was reduced in these neurons.
So the enhanced subunit is actually gumming up the works.
In a way, yes. And this impairment led to the accumulation of something called K48 polyubiquitin. That's a key marker, like a tag, for dysfunctional protein degradation. The cell is failing to process specific waste properly.
And this wasn't just in lab dishes, right? They saw this in the EAE mice, too.
Yes, they saw increased K48 polyubiquitin in the neurons of EA mice. And again, this was reversed if they deleted neuronal PSMB8 or used the inhibitor.
So a direct link.
Yeah.
PSMB8 leads to impaired protein recycling in diseased neurons in a living animal. That's solid.
Very solid. But then the question is, what specifically accumulates because of this impaired degradation? What's not getting cleaned up? The researchers hypothesize it might be a key metabolic regulator. See, neurons typically have a low rate of glycolysis. That's a fast way to make energy. And that's partly because a protein called PFKFB3, which is like a master switch between glycolysis and another pathway called the pentose phosphate pathway, or PPP.
Right.
That PFKFB3 protein is usually constantly degraded, kept at low levels.
Okay, so it's normally kept in check.
Exactly. But they found PFKFB3 levels were elevated only in the PSMB8 overexpressing neurons and also in the EAE mice.
Ah. So the broken recycling system specifically lets this metabolic master switch, PFKFB3, build up.
That was a crucial piece of the puzzle falling into place.
Okay, so what does this PFKFB3 accumulation actually do to the neuron's energy use and its overall health? This is where it gets really interesting, I think.
It really is.
With PFKFB3 building up, the next logical step was to see if PSMB8 fundamentally changes how neurons make energy. Tracing that domino effect.
Precisely. They measured ATP production, the cell's energy currency, and they found that neurons overexpressing PSMB8, or those exposed to that inflammatory signal IFN gamma, showed increased glycolysis, fast energy production.
Okay.
But crucially, there wasn't a corresponding change in mitochondrial ATP production, the more efficient long-term energy source.
So it's a shift towards the fast, less efficient pathway.
Exactly, a definite metabolic shift. And importantly, this shift was reversed if they deleted PSMB8, or, interestingly, if they overexpressed the normal PSMB5 subunit.
Right, the one that got pushed out.
Yeah. And crucially, they showed that just overexpressing TSKFB3 by itself was enough to increase glycolysis, and deleting PFKFB3 reversed the glycolytic surge caused by PSMB8 or IFN gamma.
Okay, so PSMB8 causes PFKFB3 to build up, and PFKFB3 then forces the neuron into this excessive, faster glycolysis. But that's generally not a great thing for neurons, is it? These are long-lived cells. It sounds like a quick fix with maybe bad long-term consequences.
Well, that's exactly what they found. What's fascinating here is the downstream consequence. This excessive glycolysis actually starves the pentose phosphate pathway, the PPP.
Which you said was important.
Critical, because the PPP generates NADPH, and NADPH is essential to produce glutathione, or GSH.
And GSH is the cell's main antioxidant.
It's the primary antioxidant defense.
Right.
So, predictably, PSMB8 and PFKFB3 overexpression, or IFN gamma exposure, it reduced both NADPH and GSH levels.
Wow. So the cell is basically sacrificing its long-term defense system, its antioxidant shield, for a short-term, maybe inefficient energy boost.
That's a good way to put it.
So the neuron's internal security system is being undermined. It's becoming vulnerable. And they saw this in living animals, too.
Yeah.
Not just in the dish.
Yes, absolutely. They used a very cool technique, MALDI mass spectrometry imaging in the EAE mice. It allowed them to visualize molecules directly in the brain tissue. They confirmed reduced byproducts from glycolysis, as expected. But also, crucially, they saw elevated GSH and NADPH in the neurons where PSMB8 was deficient.
So removing PSMB8 boosts the antioxidant levels back up.
Yes. It provides really strong in vivo evidence that neuronal PSMB8 drives higher PFKFB3 activity, pushes that metabolic switch, and depletes crucial antioxidants right there in the diseased brain.
Okay, so for you listening, this means a neuron under inflammatory stress because of this PSMB8 issue is essentially prioritizing a less efficient, potentially damaging energy pathway. And at the same time, it's losing its ability to fight off damaging oxidative stress. It really is like draining your car's oil to fuel the headlights. You get a bright light for a moment. But
At a huge cost to the engine's long-term health.
exactly. Okay, so we've got this chain. PSMB8 causes proteasome dysfunction, which lets PFKFB3 accumulate. That pushes glycolysis, which depletes antioxidants. What's the final blow? What's the devastating punch for the neuron that comes from all this metabolic chaos?
Well, this raises the critical question. Does this whole metabolic switch make neurons more vulnerable to specific ways of dying, with reduced activity in that PPP pathway and critically low GSH?
The main antioxidant.
Right. The researchers hypothesized it would make neurons highly sensitive to ferroptosis.
Ferroptosis, that's the iron-dependent cell death, like rusting from the inside.
Essentially, yes. It's characterized by lipid peroxidation damage to the cell's fatty membranes.
And were they right?
They were spot on. Neurons expressing PSMB8 or PFKFB3 or exposed to IFN gamma were indeed far more vulnerable to agents that induce ferroptosis. They also showed increased lipid peroxidation, that hallmark of ferroptosis.
Wow.
And critically, this vulnerability could be rescued by using ferroptosis inhibitors.
So that's a very strong causal link. The metabolic changes directly lead to this specific type of cell death.
It really connects the dots. And what's also fascinating here, they uncovered that this PFKFB3 accumulation, when driven by inflammation, it seems to operate independently of its usual physiological regulators, things called CDK5 and CDH1.
So inflammation isn't just causing a malfunction, it's actively hijacking PFKFB3 in a completely new way,
Yeah.
bypassing the normal controls.
That seems to be the case, a novel hijacking mechanism.
Okay, this brings us to potential solutions, which is always the exciting part. They looked at human MS brains, right? And found elevated PFKFB3 in the active lesions.
Yes, correlating the findings back to the human disease.
So could targeting PFKFB3 directly be a way to protect the neurons, a potential therapy?
Well, the findings here were quite compelling. They treated the EAE mice with a PFKFB3 inhibitor, a compound called PFK158, and it significantly reduced disease severity. It protected against neuronal loss, synaptic loss.
Kept the connections intact.
Yes. It lessened oxidative stress and lipid peroxidation, and even improved mitochondrial health.
So hitting that metabolic switch directly helps.
It does. And importantly, even doing a neuron-specific deletion of PFKFB3, genetically removing it just in neurons, had similar neuroprotective effects. And this was without altering the overall immune response.
Ah, that's key. It shows you're protecting the neuron itself, not just calming the inflammation around it.
Exactly. It functionally anchors the pathway, showing that targeting this specific neuronal mechanism is protective.
So for you, this isn't just, a complex diagram of a pathway. It's potentially a therapeutic roadmap. We're talking about hitting targets right inside the neuron that could protect it from the damage in MS, even if inflammation is still happening elsewhere.
That's the exciting implication. It's a fundamentally different approach to neuroprotection in MS.
Yeah, this paper really does shift our thinking, doesn't it? It starts, as you said, with this crucial unmet need, protecting the neurons themselves, not just focusing on the immune cells. It's about looking inside the target cell.
Absolutely. And the innovation here is laying out this precise cascade. Proteasome dysfunction leads to metabolic change, leads to cell death. The trigger is IFN gamma-boosting PSMB8 in neurons. Then the non-intuitive step. PSMB8 impairs proteasome activity.
Right, the opposite of what you'd expect.
Which creates this metabolic lesion, letting PFKFB3 pile up that forces excess glycolysis, starving the pentose phosphate pathway.
Are an antioxidant generator.
Leading to fewer antioxidants.
And the consequence, as we saw, more oxidative injury, more vulnerability to ferroptosis. But the real breakthrough for me is what they call causal closure. When they actually went in, genetically or with drugs, and dialed back PSMB8 or PFB3, the neurons survived better, and the disease outcomes improved in the animal models.
Exactly. It's not just correlation. It's showing cause and effect. This is what drives the damage.
It really reframes the immunoproteasome, doesn't it? Not as this general immune helper, but as a potential metabolic saboteur, specifically in neurons when they're under inflammatory stress.
Precisely. Previous efforts in MS neuroprotection often looked at mitochondrial failure or excitotoxicity or iron biology almost as separate problems. This paper connects those dots, tracing them back upstream to this cytokine-induced proteasome switch that completely reprograms the neuron's metabolism. It offers a unifying explanation.
So what does this mean for the road ahead? What are the challenges, the opportunities this opens up?
Well, there are definitely challenges. Developing PSMB8 drugs that can get into the brain and are safe for long-term use. That's tricky, given the known risks of general proteasome inhibition in cancer therapy, for instance.
Right, you need neuron-specific targeting, ideally.
Ideally, yes. And also, generalizing these findings fully to progressive human MS is still a step. But the core insight is profound. Neuroprotection isn't just about, say, patching up mitochondria after the fact. It's about controlling this proteasome metabolism link. It offers a really tractable path, potentially for combination therapies alongside existing immunomodulators.
So tackle the inflammation and shore up the neuron's internal defenses.
Exactly. And it really highlights the need to investigate these neuron-specific mechanisms. They clearly differ from dividing cells, like immune cells. Future studies should definitely explore novel technologies, maybe things like targeted protein degraders or biodegraders to specifically hit PSMB8 or PFKFB3 just in neurons. That could offer more precision.
Fascinating. This deep dive has really shown us that what might be immunoprotective in one cell type can actually be neurotoxic in another. The immunoproteasome, the system we thought we basically understood, reveals this startling cell-specific vulnerability. It's a powerful reminder of biological complexity.
Indeed. And it leaves us with a really compelling thought, doesn't it? In the incredibly complex landscape of neurodegenerative diseases like MS, could the real key to protecting our most vital cells lie not just in broadly targeting the inflammation, but in understanding and selectively tweaking these hidden cell-intrinsic metabolic pathways that inflammation subtly hijacks?
It truly changes how we might think about cellular defense and opens up completely new avenues for neuroprotective strategies.
Absolutely.
Definitely food for thought. Thank you for joining us on this deep dive. We hope this exploration has given you a shortcut to being well-informed on this and maybe spark some new ideas. Until next time, keep digging, keep questioning, and stay curious.